Welcome to the Metabolic Link, a podcast that explores the common thread of metabolism in health and disease. This is where science meets society. Welcome back to another episode of the Metabolic Link. I'm your host, Dom D'Agostino, and I am thrilled to have Dr. Robert Lustig joining me today. And Dr. Lustig is a professor emeritus of pediatrics in the Division of Endocrinology at the University of California, San Francisco. He specializes in the field of neuroendocrinology with an emphasis on the regulation of energy balance by the central nervous system. His research and clinical practice have focused on childhood obesity and diabetes. Dr. Lustig is an undergrad of MIT and achieved his MD from Cornell University Medical College. And then he did a master's of studies in law from UC Hastings College. And he then went on to do a number of different academic pursuits, including his book, which I am a big fan of, Metabolical. And I have found him to be an amazing encyclopedia of knowledge on a broad array of topics. And I'm excited for you to listen to this podcast. Please enjoy and share. Thank you. Dr. Robert Lustig, thank you for coming on the Metabolic Link podcast. I am a huge fan of your work, and it's great to have you on again.
Vice versa, Bob. Vice versa.
You're like an encyclopedia. Yeah, I always view this as like a really grateful opportunity to learn from you. And I have so many questions, especially regarding a recent review that you published with colleagues, Obesogens, a Unifying Theory for the Global Rise in Obesity. And this was published in the Journal of Obesity, the International Journal of Obesity. And with a colleague that I'm familiar with the work of Barbara Corky, right? And I was not familiar with the first author, Gerald Handel.
Jerry Heindel. So Jerry used to be the program director for endocrine disruptors at the National Institute for Environmental Health Sciences. He's now retired from that, but has taken on a executive role in a nonprofit called HEEDS, which is Healthy Environment Endocrine Disruptor Strategies.
Okay. So yeah, endocrine disruptors, which really ties into kind of what we're going to be talking about. And for our listeners here, I highly recommend for our listeners to access the International Journal of Obesity. This review article, I spent a good part of the weekend not only reviewing the article, but also highlighting many of the references in the article that many that I was not aware of. And I think it's a very well referenced review. And it's really a nuanced, integrative, unifying review that discusses, which is there's a dichotomy. Everybody knows there's this dichotomy between the energy balance model and the carbohydrate-insulin model. And they've
been duking it out. And I will tell you, I mean, I was part of the duking for a while, but when Barbara, presented this concept back at this Obesogen meeting that we held at Wingspread in Racine, Wisconsin in 2022, it became very clear that, we were all sort of working at common purposes. And so we put this review together. It took over a year.
It really does bridge the gap. And it also brings in, another interesting, theory, the redox theory that ties into accelerated reactive oxygen species and reactive oxygen species as I think Barbara Corky showed that they can stimulate insulin secretion, and they are, indicative of mitochondrial dysregulation, mitochondrial dysfunction. So it ties in that, but the obesogens, I think it's important that kind of sets the stage. So, I'm maybe putting, I don't want to put words in your mouth, but I think it kind of sets the stage for the unifying theory here, where you have the energy balance is just energy surplus, really. And then carbohydrate insulin model, you get the sustained insulin release. But in the context of these obesogens, the obesogens really feed and validate the redox model, which is excess reactive oxygen species, more or less is just pushing the throttle on this
Right.
whole thing. So
You know,
it's a beautifully written article, and I think I encourage people to pull, because I've never really read anything that unifies this all together, and I think we need this in the field. I think it's very important.
well, thank you. Let me also mention that Bruce Blumberg, who's an obesogen maven at UC Irvine and has done all this work on tributyltin and the fact that, you can show fourth generation effects after exposure to tributyltin. And by the way, we're all exposed to tributyltin because it's the stuff they paint on the hulls of ships to prevent barnacles from attaching. So they are, throughout our water supply. Bruce wrote an op-ed to our review in the journal Endocrinology called "Do We Need a New Unifying Theory for Obesity?" and was very complimentary because, after all, he's in this boat too.
Yeah.
So
that's great.
People can look at Bruce's assessment, and I think we'll learn a lot from it.
Yeah.
So here's the lay of the land, if you will. All right, everybody and his brother thinks that it's all about calories. Calories in, calories out. You eat too much, you exercise too little. Okay. Therefore, if you're fat, it's your fault. Therefore, diet and exercise, except it doesn't work. And it hasn't worked.
In practice. Yeah. I mean, there's some theory.
To be honest with you, not really even in theory. And the reason is because you can't measure a calorie. Because it's not a calorie that passes your lips. It's a calorie that it passes your intestine. And you don't know what that is. Why do you think that Kevin Hall's famous paper now is so important? It's because it doesn't matter what the calories are that you consume. It's the calories that you absorb. And
their new model, but their new model does, as the carbohydrate-insulin model does not really talk about or incorporate the microbiome much. So the last couple papers I was reading with the energy balance model, it goes into, ultra-processed foods on brain and energy regulation from that, but it does kind of expand that, I think, on the microbiome, but it has a lot of fuzzy takeaways from it. So they're just, there's kind of like, and the microbiome's involved kind of thing. Yeah.
No, if you look at either model, the energy balance model of calories or the carbohydrate insulin model of, refined carbohydrate / sugar and, insulin response, they are both inadequate. They both have holes in them.
Okay? I think we all agree on that. Yes.
Different holes, but holes. Yeah. Right. The carbohydrate insulin model doesn't explain the epigenetics or the developmental programming that occurs before birth. So either one is lacking.
And obesity starts in utero. That's stated at least twice in the article. I remember because I went to the references to read up about that. And I think that's really important to acknowledge. And
four separate studies, US, South Africa, Israel, Russia, the secular trend of birth weight over the last 25 years has been an increase of 200 grams. Okay, babies weigh 200 grams, a half a pound more today than they did 25 years ago. Now, you might say, well, then they're healthier, except that when you put them in a DEXA scanner to see what those 200 grams are, it's fat. So they are 200 grams more obese than they were, and they were fetuses. So you can't do carbohydrate insulin because they didn't eat anything. And you can't do calories because they don't diet and exercise. Okay? So neither of them work.
But wait, does that take into account the BMI or the body composition of the mother? So is that standardized to that?
Like, it's not standardized in those papers, but the fact of the matter is, if the baby ends up 200 grams more obese, it really doesn't matter whether the mother was more obese or not. It is true that the more obese the mother, the more hyperinsulinemic the mother, therefore the more glucose gets across. That's true. The question is
that's how I think about it. I think about obesity, like the epigenetic patterns are set with the mom, and ultra processed food, activating epigenetic programs that confer obesity phenotype for the offspring. That's kind of how my simplistic mind thinks about it.
So what we're saying in this paper is, what is the epigenetic aspect of it?
Yeah.
That's what we're saying. All right, so let's discuss what this thing is, okay?
I want to take a brief break to say a special thank you to Genova Connect, who is a sponsor of this episode. Genova Connect, powered by Genova Diagnostics, offers a simple at-home advanced lab testing that's grounded in research and takes the guesswork out of our understanding of health. Genova isn't just a lab, they're innovators in health and wellness. Whether it's gut, hormonal balance, nutritional insights, their tests provide clarity and direction, allowing you to optimize your metabolic health. I originally found out about Genova from a patient that emailed me years ago and sent me their report. And I was really, really blown away by the breadth and depth of the report and the details that it went into in regard to sort of the metabolic pathways, and specifically relating to the neurological disease of this patient. I found it very insightful and also actionable. So Genova Connect is now offering you a 15% off of any of their test kits when you use the code Metabolic Link at gdx.net/the metabolic link. Now let's get back to our episode with Dr. Lustig.
First of all, obesogens don't have calories, but they drive adiposity. And the question is why? Well, they can do one of two things. They can either bind to receptors that drive adipose tissue, like, for instance, the estrogen receptor or the glucocorticoid receptor or, the aryl hydrocarbon receptor. Okay. And so they don't have to have calories, but they'll still make fat.
Mm-hmm.
Or they can do this redox trick. Okay. So as everybody knows, redox means oxidation reduction. Okay. And it has to do with the generation of reactive oxygen species. Now, everyone in the obesity field knows what reactive oxygen species are. They are what are given off in the process of energy metabolism. And if they are not quenched, they do damage. Okay. It's one of the primary reasons for metabolic syndrome. It's one of the chief reasons for non-alcoholic fatty liver disease that progresses on to inflammation, because reactive oxygen species that are not quenched can peroxidate lipids, it can denature proteins, it can, that's why we have this thing called the endoplasmic reticulum stress, which is also known as the unfolded protein response. They are the same thing. So reactive oxygen species are made in the mitochondria. They are made in the cytosol, from normal energy metabolism, and they are supposed to be quenched in the peroxisome, because the peroxisome is where the antioxidants are. That's where the glutathione is in the liver, and the vitamin E and vitamin C and what have you.
And NNT too. So I'd like for you to, because this is near and dear to my heart, because there's a C57 Black 6J and the C57 Black 6NJ. And we saw that the J, which lacks NNT, they died real quick with a tiny dose of LPS. So we did some LPS studies for inflammation, and they also present with unique metabolic syndrome. And, I went down the NNT rabbit hole, which I've done many times because we have an expert in our lab that got her PhD on this subject. And I think the NNT, that kind of story, the redox story, kind of lends itself. There's a lot of really nice experimental animal model. So maybe because that's in the model of the review article, and it's kind of, I kind of hyper focused on that. So that's really part of the model.
So bottom line is, everyone knows that ROSs are bad for you. Well, not really. Depends on whether or not whether they do damage. So in excess, in high excess, yes, they will do damage. But what the paper is saying is that even when they're not in excess, they're part of the energy control system of every cell.
Signaling. Yeah. ROS are very powerful signaling molecules.
It's an autocrine signal. It's a signal for inside the cell. So think about it this way. Every cell has the capacity to grow, and it has the capacity to burn. And at any given point in its life cycle, it will be called on to grow, or it will be called on to burn. And the question is, what's the switch? When does growth become burning? When does burning become growth? And the answer is these three enzymes in every cell determine which way the cell goes. Those three enzymes are PI3 kinase, phosphatidylinositol 3 kinase, which is the spigot that opens up, increased glucose coming into the cell. This is the enzyme that Lou Cantley, discovered many years ago. And
my postdoc was trained by his, Gina DiNicola, who was his protege. Yeah. So that pathway is very near and dear to ours. Yep.
That's the cancer gene and protein, because it allows for more glucose entry, because after all, cancer cells don't use mitochondria to create ATP. They have to use glycolysis because they need the pyruvate for structural components instead of for burning all the way to carbon dioxide. So PI3 kinase, AMP kinase, which is the fuel gauge on the liver cell. It's the one that tells the cell to make more mitochondria because running out of energy. And the last one is mTOR, mammalian target of rapamycin, that tells the cell basically to divide. It's time to grow. So when these three enzymes, which by the way are all kinases, are in sync with each other, the cell will grow. And when they're in opposite sync, they will burn. All right? That's the way this works. What tells those enzymes whether they are in sync or not is the degree of reactive oxygen species, because ROSs ultimately lead to alterations in phosphorylation status of kinases. And so those kinases will then change their polarity, their valence. And so we'll go from importing glucose to not importing glucose, or, stimulating mitochondria to not stimulating mitochondria. And they all do it presumably in concert. And so this is the energy switch inside the cell, and it's run by ROSs. So the beauty of this theory is that if something mimics ROSs or generates its own ROSs having nothing to do with energy, you can get changes in those three enzymes, which will then dictate an energy metabolism path that maybe the cell didn't want originally. Okay.
And by virtue of the mitochondria being the primary source of ROS, if you take a step back, you're talking about a mitochondrial disruptor that, you have NADPH oxidase and other sort of, xanthine,
that are outside.
But the mitochondria for the most part is, 90% of the ROS production. ROSs. So you're talking about
absolutely
a mitochondrial, disruptor. Yep.
So basically anything that disrupts the mitochondria is going to generate more ROSs. And so that's going to take the cell in a completely different direction in terms of what it's going to do in terms of either energy burning or energy storage / growth than it would have otherwise.
And that's sort of in line, I think, with the energy balance because, a surplus amount of calories lead to, well, in their model, the calories then promote adiposity or whatever, but surplus amount of calories are amping up ROS production, which desensitizes and disrupts those signaling enzymes and kind of sets the stage for that. But and maybe, you could talk about the obesogens and maybe the ones that could be most important: the phthalates, the bisphenol A, the glyphosate, and there's a number of high fructose corn syrup
of them.
And drugs too.
Right, absolutely.
Non-typical antipsychotics are highly metabolically dysregulating. Yep.
Exactly right. And so the question was, how do they fit into, either the calorie model? They don't, or actually the carbohydrate-insulin model, and they sort of don't either. Well, we know that some of them cause insulin resistance, like for instance the atypicals. Yes, that's true, but it still didn't explain why the pancreas would basically go hog wild. Well, we now know, because ROSs in the pancreas, basically what that's saying to the pancreatic beta cell is, got too much energy here, gotta store it. So let's release the insulin in order to store it. And so that explains the hyperinsulinemia that we're seeing in all of these energy
Mhm.
surfeit models. So,
Yep.
and the thing that's really cool is that different, different organs, okay, manifest the ROS excess in different ways. So in the brain, it manifests as increased hunger. In the pancreas, it manifests as increased insulin. You know, in the
Mhm.
liver, it manifests as increased de novo lipogenesis. You know, all basically to deal with this notion that there's too much energy in that cell, and the reason is because those three enzymes flipped. So
Yeah.
it's actually a very elegant model, and it then explains why increased calories do it and why increased carbohydrates do it, because carbohydrates generate ROS way faster than fat. That's one of the
Yeah.
reasons ketones are, quote, good, unquote, is because they don't generate nearly the amount of ROS that carbohydrates do. So
Mm.
it's internally consistent. And then, of course, it explains the obesogen story, which
Yeah.
everybody has ignored because it didn't fit either of the two models, but in fact is extraordinarily important and was actually the window onto how to explain this. And that's what we figured out at this wing spread meeting, which was really quite good.
Yeah, and it doesn't really discredit either of the models. They're important contributors, but the models are presented in a way that they're mutually exclusive, and this creates a dichotomy and confusion in the field. And I think it's a much more nuanced, approach to that. These are overlapping. That there's multifactorial. There's other contributors. And that ultimately are what you're talking about is like really setting the stage for these models to exist. That's why we're talking about them.
Well, what it
If it's
also
just simply
does,
energy, it's not. Yeah.
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 Well, what it also does, and we make a very important point of this, in the paper, especially at the end, is that it actually provides a window into what to do about it.
Yes. Hmm.
The other two don't. What are you going to do about
Yeah.
calories? Really? You can tell people
Yeah.
eat less. Yeah, really. That's worked really well, hasn't it? Okay. What are you going to do about carbohydrate? Yeah. Okay. Well, so the ketogenic diet, but not everybody can stay on the ketogenic diet.
Yeah.
And I will tell you, and as much as I value the ketogenic diet, as much as we use the ketogenic diet in our clinic at UCSF, okay, the fact of the matter is that when people do the ketogenic diet without physician input and without monitoring, within 2
Yeah.
months, they've regressed to the mean and they're not on a ketogenic diet
Yeah. Hmm.
anymore. As soon
Yeah.
as you introduce carbohydrate in and the insulin starts flowing, okay, ketogenesis is over. So
And you could rebound too from that too. And
absolutely.
yeah.
And if
Hmm.
you're not, and if you try to be on a ketogenic diet, but you're not, then you are on a high fat, medium carbohydrate diet with insulin. That's actually the worst diet to be on for chronic metabolic disease and cardiovascular risk. So to me, um, the beauty of this redox model is it tells you what you need to know to try to prevent it. Okay. So number one, okay, all the things that generate ROSs, which is a lot of stuff, is potentially an issue. But the thing that generates ROSs the most, without question, without, any, issue is ultra processed food. And that's what
Yeah.
we now know is the big problem. Ultra processed food is basically ROS in a bottle or in a pouch.
I think we should also emphasize like the maladaptive response to sustained ROS and ultra processed food is doing that through an energy surplus. And then because there's exercise, right? So the biggest, stimulator of ROS is probably, exercise, but the adaptation to that stimulus ultimately improves mitochondrial function and therefore will decrease ROS over time. So I think it's important to talk about maybe some food specific obesogens, and then maybe talk a
Well,
little bit about like Teflon and the
yep,
PFAS,
PFAS,
thing.
PFAS
I
is
mean,
right.
I think I kind of thought it was a little bit of fear mongering until I kind of delved into the research a little bit more and realized that yes, these
No,
are
it's
these are
real.
contributors. Yeah.
Oh no, they're real. They're
Yeah.
absolutely real. The data is very clear. And in humans too. I mean, obviously we don't have randomized controlled trials, but we have
Yeah.
econometric analysis to demonstrate that. Here's the reason why ultra processed food is, ROS in a package. Okay. And it's something that people have ignored. Dietary advanced glycation end products, DAGEs. So everybody knows about AGEs. Okay, glucose binds to a protein that generates the Maillard reaction. Throws off a reactive oxygen species, and also now you've got a cross-linked protein. That's all true, and I don't take anything away from that. That's a huge problem. Okay, but dietary AGEs. So you take carbohydrate with protein, okay, and you heat it. All right.
Yeah.
And it can be your, your cake, or it can be, the stuff, you buy at the grocery store, the McDonald's, the Hostess, cake, or the one you make at home. Doesn't matter. The point is, dietary AGEs, so, the thing that caused these things to brown, okay, you consume them, and people thought, well, it's not a big deal. Well, those dietary AGEs bind to a receptor. Receptor for AGE is called RAGE, RAGE. Okay, and that RAGE, that receptor, is coupled to NADPH oxidase.
Yep.
So you are generating ROSs directly from the food you eat, having nothing to do with its calories. So it basically says ultra processed foods going to be obesogenic because of the increased ROS burden that it's going to place on cells. So it tells us what we need to do in order to be able to fix that. And of course, Kevin Hall's paper shows that processing matters. Indeed, processing does matter. It's not even what's in the food, it's what's been done to the food that matters more. And so those dietary AGEs play a major role if you, subscribe to this redox / obesogen model.
I want to take a few moments to say thank you to Element, who is a sponsor of this episode. Electrolyte deficiency or imbalance can cause headaches. It can cause cramps, fatigue, brain fog, and weakness. Element can help you stay hydrated without the sugar and other dodgy ingredients found in the popular electrolyte drinks and sport drinks that you find on the market. Element is it's a zero sugar electrolyte drink mix, and it's born from a growing body of research revealing that optimal health outcomes occur at sodium levels that are 2 or 3 times the government recommendations. And this is true even more so in those that are keto or on low carb. I've been using Element for at least 5 years now, and I love the product and I love the taste. Element is offering for our listeners an exclusive deal. Get a free Element sample pack with any drink mix purchase when you visit drinklmnt.com/metabolic link. Now let's get back to our interview with Dr. Lustig. Thanks. Yeah, and the ultra processed food really ties into the carbohydrate insulin model too.
Yeah.
Just making those carbohydrates, not even necessarily sugar, immediately accessible to, the upper duodenum, which they probably just get sucked up so quick. It's like a bolus of sugar hitting the bloodstream, and it doesn't even have to be liquid calories, which is probably the worst offenders, sugar sweetened beverages. But yeah, it sets off a signaling cascade unlike anything that we've probably experienced sort of in the past. And maybe even if it's not in a protracted way, it's stimulating an energy surplus and the ROS production within a sort of a compressed time frame, because you could even eat that stuff and then fast and eat that stuff, but you're still kicking on that machinery. Yeah,
absolutely. And it can last a while.
Yep.
So the good thing about this, shall we say, unifying hypothesis is that it, number one, doesn't throw either of the previous models out. They're both inclusive. It's inclusive of those, and it also plugs the holes, and it explains why things that are not caloric still cause obesity, which I think is extraordinarily important. People, haven't wrapped their heads around this at all yet. people are
like
still
insulin's not caloric, but causes rapid weight gain, right? So,
no kidding.
but you can imagine highly individual susceptibility to different aspects of the model. And I think that
Sure.
maybe opens itself up to testable hypotheses. And I know in the latter part of the review article, I think you kind of talk about maybe research that should be done or needs to be done. So I think maybe we could talk a little bit about, that research, but also how do we, I mean, because some people, I know quite a few people eat processed food and they're skinny, so they somehow auto regulate, or that now, who knows what's going to happen 20, 30 years later. But some people can eat ultra processed food and be totally fine, whereas some people are highly susceptible to it. And, if they transition from one type of diet to another, you can kind of see that. So,
Right. I understand that, and I agree, and it depends on what the ROS burden is. Well,
yeah.
actually, you can measure that because there are, markers for that you can measure in blood and
Mhm.
urine to tell you whether that's actually the case. So,
What are
this
the best
requires,
markers, Rob, that I mean, we've done
oh,
T-bars tests and malondialdehyde
yeah,
and, different.
malondialdehyde's a
Yeah.
good one.
Yep.
Yeah, that is probably the best.
Okay.
It's the
hmm
easiest to obtain,
hmm
probably the cheapest too. The
Yep.
point is, you could set up a research project with a, specific diet, that
hmm
basically is a low ROS producing diet. And the thing that basically, gets rid of the ROS is sulfur. Okay.
hmm
So you have to have cysteine and you have to have methionine. And we know that cysteine and methionine are in low concentration in ultra processed food. So you could take an ultra processed food diet and supplement it, say, with cysteine and methionine and see whether or not it makes a difference. The methionine being the methyl
hmm
group donor for glutathione.
Yeah. Like N-acetylcysteine, NAC, or
Right.
something like that
Yeah.
thing. Okay.
Right. And people are
Uh-huh.
using NAC to try to reduce ROSs for all sorts of things, including
Mhm.
psychiatric diseases like
Yeah.
OCD, for
Yeah.
this
It's a
reason.
standard of care, I think, for acetaminophen toxicity or something, right? NAC.
Absolutely. Yeah. Yeah.
Yep.
Right.
Hmm.
Mucomist, we in
Yeah.
pediatric parlance, that's what we give Tylenol for.
Yeah, that's right. Yeah, it's like nebulized, right? Or
Yeah,
yeah.
nebulized
hmm
Mucomist, right?
Yep. So,
Exactly.
but there's plant constituents that could also be. I've been going down the rabbit hole of reading about urolithin A. I think it's sold as
Yeah,
Mito
yeah,
Pure, and just
yeah.
started testing that on myself. And that's in very high concentrations and the stuff that are really good for you, like berries, raspberries, walnuts. I think are really high in those things. So maybe incorporating specific types of foods,
Mhm.
can help reduce that ROS burden and enhance, sensitivity in that way.
The point is
Insulin.
that this is a hypothesis, it's a
Yep.
model. Okay. It is by no means fact at this point. It needs to be tested. But you can't test a model till you've thought of it and
Yeah.
and gotten some buy-in. So what I'm hearing from you, Dom, after reading this is, you're bought in, which is
Yeah.
good.
hmm
Now we need
Yep.
to now we need to, get enough, investigators on board so that we can actually, do this as a multi-center
Yeah.
evaluation slash trial.
One of the biomarkers, I think, because ROS contributes to inflammation, right? There's IL-6, IL-1 beta, TNF
Yeah.
alpha, maybe just a generic one that seems to be even the metabolic psychiatry researchers are focusing on a simple one like HSCRP, right? And maybe that could be, and I know HSCRP correlates with waist size. So the higher the waist size, the more HSCRP, and that would be insulin resistance and things.
Right.
So maybe there's some simple biomarkers that we need to, not that we're not kind of doing that already, but I think
Yeah.
maybe a
Yeah.
panel that would could be used in helping to validate this. And
Right.
maybe it's and also epigenetic, I think, looking at epigenetic patterns. I got my GB Health Watch back, and looking at I have a missense mutation in the MPC1L1, and so I'm a hyperabsorber of cholesterol, and that's why a tiny microdose of ezetimibe knocks my ApoB in half. So I think there's some interesting insights I think that we could do from some genetic testing, and
That's
but
interesting. I
yeah.
didn't. I didn't even know that that was a thing. So thank you.
Yeah, yeah. Well, we absorb, fat and cholesterol in different ways. And I always, just teach, my cells, absorb fat and cholesterol, but there's this MPC1L1 receptor, and that impacts and maybe, I mean, this goes into processed food and stuff too. I think it's kind of all tied together on our lipid profile, but I think it'll be a constellation of certain biomarkers I think that we need to look at to and develop the testable hypotheses. And the question is, what's the question? We always say that in the lab, and then we build our hypotheses on that. But I think you have a compelling, very provocative review here that needs attention.
Well, thank you.
And by researchers,
Well,
yeah.
I have no doubt Jerry and Barbara will be very delighted to hear, that it's, you got some traction.
Yeah.
We obviously, we need to, sort of broadcast this far and wide. And so I'm very happy that we're doing this,
Yep.
podcast now. Very specifically get this out to the public and, let people take their pot shots at it.
Let's take a break to highlight and thank AquaTrue, a sponsor of this episode. Having safe, clean water is the last thing you want to worry about. But unfortunately, according to extensive research by the Environmental Working Group, 3 out of 4 homes in America have harmful contaminants in their tap water. That's why you've got to check AquaTrue. AquaTrue purifies using a 4-stage reverse osmosis purification process, and their countertop purifiers work with no installation or plumbing. This unit will remove 15 times more contaminants than the ordinary filtration systems that you find on the market, and are specifically designed to combat chemicals like PFAS. These are the polyfluoroalkyl substances that you hear about in nonstick pans, and the units will filter them to get them out of your water supply. So AquaTrue comes with a 30-day money-back guarantee and even makes for a great gift. Today, our listeners will get a 20% off of any AquaTrue purifier. Just go to aquatrue.com. That's AQUATRU.com and enter the code Metabolic Link at checkout. That's 20% off any AquaTrue purifier. And if you go to aquatrue.com and use the promo code Metabolic Link, METABOLICLINK, please use that code at checkout, and please enjoy the rest of our interview with Dr. Lustig.
I just received an email today from an Iranian reproductive medicine physician who had questions about it. He said, and I don't know the answer to this, but he said, yes, oxidative stress is important, and yes, reactive oxygen species are important, but there's also something called reductive stress, which also leads to changes in the ROS pool. I would think it'd be the opposite, but apparently no, it doesn't. It's it can cause its own problems. And so how does that work? And so they're sort of my brain exploded just before coming on the podcast today going, all right, what am I going to say about this? I don't know.
Yeah. Yeah, someone brought that to my attention, I think, because beta hydroxybutyrate, or at least the R enantiomer, can have a reducing effect through different metabolic intermediates. And it sent me down that rabbit hole. Redox biology, I teach it, and it's very complex, and I don't understand it. But
indeed. And I think that's one of the reasons why, this sort of hadn't been put together before.
Yeah.
So, kudos to Barbara, for bringing this to our attention. And then when we started assembling all the pieces, because she didn't know a lot about obesogens, but boy, oh boy, Jerry sure did, being the endocrine, head of endocrine disruptors at NIH for, decades. And he wasn't, when he started, he was not enamored of the idea that endocrine disruptors could be obesogens. But now, you do the research and, you change your mind.
Are there any home tests for these things? I think I've tested our water for I know a carbon water filter takes out maybe some of the phthalates and maybe bisphenol A and things like that. But
I don't know of a home test yet. Obviously you can test large reservoirs, professionally, and you can do GC mass spec and find all of these things, and they're all quantifiable. But I do not know of a home test for obesogens as of yet.
So I think about the phthalates as being like, and we use all this plastic, wrapping for things, and we're heating things up in plastic. So I think of that being a contributor. Then there's different tests for like glyphosate and other, that's a herbicide, but different pesticides and stuff too. So I get a lot of questions about this. What I do, I test heavy metals. I use the Genova Diagnostics. They have some good tests for like heavy metals. I've done like the hair analysis and things like that. I live on a farm, so kind of, there was a lot of pesticides and herbicides and many different chemicals of things that I wasn't even aware of that I know are in the soil and stuff too. Although we tested it and it was kind of clean, but I'm always just curious about these things.
So, something I might mention here. Okay. One thing that we know drives adiposity is estrogen. Okay. Yep. And estrogens are everywhere. Okay. And the reason is because the estrogen receptor is the most promiscuous receptor. It'll bind to anything. Okay. If you have two hydroxyl groups 22 angstroms apart, you're an estrogen. Okay. And so the various things that are estrogens in our environment is myriad. I mean, there are, columns and columns of, chemicals that we're exposed to. But here's the thing. Estrogen, when it binds to its receptor, it basically changes cell physiology and promotes stem cells turning into adipocytes, for sure. No question there. That's one of the reasons why girls get curves at puberty. Yep. But where do they get their curves? They get their curves in sub-Q fat, not in visceral fat. Okay. Because sub-Q fat is where your body wants to put energy. That's where it wants to store energy. That's your, emergency spare tire, if you will, is your subcutaneous fat. Well, subcutaneous fat does not generate a lot of ROSs for the body. And so subcutaneous fat, while sometimes cosmetically undesirable, is metabolically inert. And so you can develop adiposity, but not necessarily disease. However, if you're generating ROSs due to obesogens because you're mimicking things that generate ROSs, like, for instance, fructose or alcohol is a good one. That's a specific ROS generator. Even though it's caloric, it's a specific ROS generator, and nobody has any doubt about that. Okay. That's going to cause more damage because it's generating ROSs in cells that aren't designed for energy storage, like, for instance, the liver, like, visceral fat. It's not, you're not supposed to be able to put so much in there. So, and that's why visceral fat and liver fat are worse for metabolic disease than estrogen is, which actually protects against metabolic disease. That's one of the reasons why premenopausal women are actually protected from fatty liver disease, protected from cancer, until, of course, they become postmenopausal, and then they catch up with the men. So, this redox hypothesis actually explains the difference between the sub-Q and the visceral and liver fat as well. So it's really a very elegant and all-inclusive hypothesis when you take it apart and examine each of the components and think about how it works. Now, the question is, can we get people, shall we say, reorienting around this? And I hope so. I mean, I hope you will help.
Yeah, absolutely. I have a provocative question. What if there are obesogens in healthy plant-based foods, right? I get a lot of questions. Yeah, goitrogens, things that decrease, thyroid or, suppress.
Right.
So one could, speculate to, well, there's anti-nutrients and stuff too that play a role.
Lectins,
lectins, yeah.
Right.
So that wasn't in the model. That wasn't in the paper, I don't think, but does it fit with the model
we didn't put it in the paper, but it fits the model.
Okay. I mean, However, I mean, and some of these things kind of are in high fiber foods. And I've found listening to you and reading Metabolical and stuff, incorporating more fiber in. But for me, I do really well on like, I guess, moderate to low, like not moderate fiber, not high fiber. So
Right.
yesterday I started tracking and I had a pound of broccoli, a cup of walnuts, a cup of blueberries, and one apple. And that was exactly 25 grams. It sounds like a ton of fiber, but it was actually 23 grams. It
Well,
sounds like a ton of fiber.
that's a good amount.
Yeah, yeah. But that was a high day for me. And I probably get about half of that, which is maybe like 10 to 15. But
Well,
I
you
feel
probably
like
get
that's
more than that.
sufficient.
You probably get more than that because you watch your food, and most people
Yeah.
don't watch their food. And the median consumption of fiber in the United States is 12 a day. So
Okay.
if you're Dom D'Agostino, you're probably
Hmm.
eating more than 12 a day on a normal day when you're not trying to push it.
Hmm.
That'd be my
Yeah.
guess.
I was actually surprised that 1 pound of broccoli is 11.6 grams of. I was thinking, okay, that's my 30 grams right there. But when I do, when I pull the different, like the USDA charts and things like that, I was like, it's got to be more than that. I always thought, because some days I'll eat like no fiber, like just meat or eggs, fish, meat, and I'll just have a pound of broccoli and be like, okay, I have like twice the amount of fiber, but that's like 11 grams of fiber. So I think so I could see why most people are just kind of low on the fiber scale. And fiber, just wearing a CGM, when I eat protein with the fiber, that has a much more balanced response. My insulin has went down, hovers
Yeah,
around 3 instead of 4 now, just by adding fiber.
no
Yeah.
question.
Didn't change
Because
calories at all.
you're absorbing it slower.
Yeah.
You
Hmm.
know this. And by the way, I'm full disclosure, I am the chief medical officer of a fiber company,
Oh, I didn't know. Hmm.
yeah, called BioLumen, and it
Uh-huh. Oh,
you know
yeah, I've heard of that company.
yeah.
Yeah. Hmm.
So BioLumen, basically, we now have a product that's on the market, Munch
Hmm.
Munch. You can find it at munchmunch.shop. I didn't do this,
Yeah.
but
Hmm.
since you brought up fiber, the point is people can increase their fiber intake in a very rational way because it's both
Hmm.
soluble and insoluble fiber at the same time. And we actually
Hmm.
have, randomized controlled trial data from both India and Australia that they demonstrate metabolic benefits
Hmm.
from consuming this. And because 1 gram of this product soaks up 6 grams of carbohydrate, you can use very little fiber. 1 teaspoon of fiber will absorb 25 grams. Basically, one teaspoon will take care of one 8-ounce Coca-Cola. That's pretty good when you think
Wow.
about
That's interesting. Yeah, I started, I forgot to mention that I started psyllium fiber because I want to wean off the ezetimibe. So I went down to just like a half a dose every other day. And now my
it. Oh,
ApoB is the same. LDL went from like 200 and something down to 100.
well, I'm glad to
And
hear that. I'm glad to hear that. But
yeah,
psyllium is soluble fiber. And
soluble. Yeah.
psyllium is not, psyllium's what's in Metamucil.
Yeah.
It's actually not good enough. And the reason is because soluble fiber, it does some good things. I'm not saying it doesn't, but there's
Yeah.
uh, uh, there's a panoply of effects that both soluble and insoluble fiber give you. And
Mhm.
insoluble fiber gives you a small number. Soluble fiber gives you a small number, but when you put the two together,
Yeah.
which is what real food is, you get all of them.
Yeah.
And so it's synergistic. And the
Yep.
reason is because the insoluble fiber forms a lattice work like a fishnet on the inside of your intestine. The soluble fiber are globular, they plug the holes in the fishnet. And so
Mhm.
together, they form this impenetrable secondary barrier in your intestine that limits the absorption of carbohydrate and fat for that matter, which is why the fiber does as well as the ezetimibe for just
Mhm.
that reason. So one of the ways to reduce your ROSs is a high fiber diet.
Yeah. And that's probably just through the gut barrier. So it's like
Yeah.
stimulating like things like akkermansia, which helps with like mucus production, maybe the Brunner's glands and stuff. We're reviewing all this in the GI section of MedPhys now. But yeah, so fiber, but it's interesting, and I would like you've probably commented on this many times because people probably ask you, because I get this question a lot, that people tell me that, and I have to agree with them that fiber is not essential. Fiber is completely unnecessary because you have people that are put on diets that are completely devoid of fiber and their health improves. So I would have not thought that, but I have kind of witnessed it, at least sort of in the carnivore community, which I'm kind of like loosely associated with in one way or another because it's like ketogenic, and ketogenic diets are kind of, historically kind of low in fiber and
Well,
can be
they don't
therapeutic.
have to be.
They don't
They,
have to be.
they
No,
don't
mine's
have to
not.
be.
Yes, they don't. Yeah.
You can eat a lot of green vegetables and maintain ketogenesis just fine. As
Yeah.
soon as you add starchy vegetables, it's a different story. I mean, a potato is
Yeah.
not going to, it's not on the ketogenic diet, but green vegetables certainly are. And in fact,
Mhm.
I think that people need, the vitamins and the flavonoids that are in green vegetables in order to make a ketogenic diet replete. Now,
Mhm.
carnivores are interesting, and I'm not going to be able to tell you exactly why it works. I mean, I know that being a carnivore, is fine. And I don't know if there's an improved vitamin and flavonoid extraction because there's no other place to get them,
Well,
or
liver has folate, it has vitamin K, it has vitamin C, it has
maybe
everything.
that's
Someone,
why.
I had to fact check this, but I looked at it. It looks like, you're eating broccoli times 10 with liver if you get that in.
What I can say is that, I came to this. I don't know if you can see it, but you see that chair over there behind me that right now is working as a storage vessel right
Yeah,
there.
I know there's a history behind that chair
That
or
yeah,
something.
that
Yeah.
chair, that chair
Mhm.
is was William or Stephenson's chair.
Oh, that's right. That's right. Yes.
Okay.
Mhm.
William
You
or
mentioned that to me.
Stephenson
Mhm.
for your
Yeah,
audience.
I've read up on him years ago. Yep.
He was a founding member of the Explorers Club.
Yep.
He didn't go to the North Pole first. He went to the North Pole most.
Mhm.
He was actually caught in in a, shipwreck
Mhm.
in
Yep.
the way northern
Antarctica.
Canada.
Yeah. Mhm.
Yeah, way at
Or the Arctic.
1914.
Yeah. Mhm. Yep.
And he ended up spending 2 years living amongst the Inuit and did fine. But what he realized was that nobody died of any disease. They were, they were the healthiest people around. And he ended up writing a book called Cancer, a Disease of Civilization, which I
Mhm.
have on my shelf here.
Mhm.
The reason I know all this is because my mother's second husband's brother-in-law was William or Stephenson, and that's how I got the
Wow.
chair. Yeah, tell me about it.
That's
We owned
awesome.
Stephenson
Mhm.
Farm in Vermont
Mhm.
for many, many years. Um,
Mhm.
the point of this is
Mhm.
that in 1928, Stephenson and his uh, uh, expedition partner from that shipwreck uh, checked themselves into NYU Bellevue and stayed there for a year and ate nothing but meat for one whole year. And at the end of the year, they were healthier than the doctors. Okay.
Yeah, it's like a
Published
superfood.
This
Meat.
was
Yep.
this was published in
Yep.
1928 in
No
Journal of
fiber.
Investigation
Yeah.
with no fiber. So I know that you can get away without it, but I can't tell you exactly why. But perhaps
Yeah.
you're right about the liver, being such a phenomenal storehouse of all of these
Yeah.
micronutrients. Maybe that's why.
Yeah. Well, it's dense in that area. And I know we're kind of coming up on time, and I know that you have a hard stop
Yeah.
coming up. But I just want to quickly mention you did mention an article about Nestle getting children hooked on
Yeah.
sugar in low income countries. So I would tell people listening to like go to your social. I think you posted it there,
Yeah.
and it's a very
It's
compelling. So maybe a couple words on
It's
that.
a nonprofit out of Zurich, Switzerland called
Yep.
Public Eye, and they've
Public
been
eye. Yeah.
eyeing
Mhm.
Nestle for a long time. And I helped them with their deep background on this
Mhm.
analysis, early on in their in their sojourns through this. But it turns out that they've sent biochemical samples for analysis uh, of various Nestle of the same Nestle product, Cellular, which is an infant formula,
Mhm.
from all over the world, and it turns out that in impoverished nations, the added sugar content of Cellular is high, whereas in the UK and in Germany, it's 0.
I saw that.
Unbelievable. The question is,
yep.
Why would Nestle be changing the formula based on the population's net worth? And we sort of know the answer.
I mean, I could see that they could rationalize it. Well, the kids have an energy deficit, so we need to put in more energy into, more calories to fatten them up or something like that. I could see them rationalizing in that way, maybe.
Yeah, well, I hate to tell you, but we're doing that in America too. There's a lawsuit.
Yeah.
A major infant food manufacturer right now over the exact same issue.
Wow.
So this is a big deal.
Yep.
So this is a window up to this.
Thank you for bringing that to the eye of the public attention, and thank you again for joining us, Rob. It's always a pleasure.
Dom, there's nothing I'd rather do than shoot the what with you.
I can't wait to drop this. I know it's going to stimulate a lot of discussion, a lot of access to your article here, and hopefully lots of shares, and get that out there and get this information out there. We definitely need it. Got to do something different.
My pleasure. My pleasure.
Thank you, Rob. Appreciate it. Till next time. Thanks for listening to this episode of The Metabolic Link. If you like this episode, please be sure to leave us a review, share it, and please drop us a comment. If you want more content like this, including access to our private podcast feed that offers exclusive ad-free episodes and also provides continuing medical education for physicians, please visit our medical education platform, the Metabolic Initiative, at membership.metabolicinitiative.com. Your first 7 days are absolutely free. Thank you again for tuning in.
Do you love learning about metabolic health? So do we. It's why we created the Metabolic Initiative, an online educational platform providing evidence-based education on metabolic health and therapies for healthcare professionals and the general public. By joining the Metabolic Initiative, you'll gain access to hundreds of expert lectures, interviews, panel discussions, and even private episodes of the Metabolic Link. CMEs are available. Go to metabolicinitiative.com to get started. And as always, thank you for listening to the Metabolic Link.